Today we're going to talk about confidence intervals, and the truth of the matter is if you understand confidence intervals, you have a very good understanding of modern statistics. Stats really do make us superhuman in some regard. So I want you to think about the process. I get a sample and I try to make inferences about a population. And let me put that into perspective. When I run a study at Regis, I get a sample of Regis students, and then I use the statistics to try to make inferences about all humans or all Americans or even all Coloradans. And the statistics allow me to make these conclusions to some degree. Now, I want you to think, let's go back to that question about niceness from a previous lecture. If I want to actually know the population average level of niceness, I would have to measure all humans on the planet. If I wanted to know all Americans' niceness, I would have to measure all Americans on the planet. If I wanted to even just measure all Coloradans' niceness, I would have to measure all Coloradans on the planet. That is something that I couldn't even figure out how to do, even with internet, right? Even with modern methods, telephone surveys. It would be an impossible task for me to collect data on all Coloradans. And I just want you to see that that is a very impossible thing for me to do on the ground level. However, my statistics allow me to make conclusions about all Coloradans, about all humans. But if you think about it, who could actually do that? Who would have the power to measure all Coloradans, all humans? The US government tries with the census and they never succeed. They don't get all humans, all Coloradans, right? So it'd have to be something more powerful than the United States government. Something like the Greek gods with these fantastical powers. Maybe your Marvel superheroes, omniscient beings, right? Somebody that knows everything, or the all-knowing God, right? This is a massive power that statistics provide us as humans. The ability to make inferences about populations that are much greater than anything we could ever measure with our own hands. A lot of my research is done online where I do get random samples of all Americans, and then I can make conclusions about all Americans, which is what I'm often doing. But without the statistics that I use, I would have to get data on all Americans. The statistics we use allow us to take a sample and make a conclusion about a larger group of people that would be impossible for us to measure. So with this in mind, stats really do make us superhuman. What is a confidence interval? In the previous example, we were talking about the superhuman abilities. Now you can imagine an omniscient being. They wouldn't need a confidence interval. They would just tell you the population value you're interested in, and it would be done. For us, we have to provide an interval that tells us we're pretty sure that the true population value falls in this range. So a 95% confidence interval, what that means is if you ran 100 experiments, 95 of your 95% confidence intervals would include the true population value that you were interested in. Now, an incorrect way to think about this is that your 95% confidence interval means that there's a 95% chance that your true population value that you're interested in is inside that interval. That's wrong. However, it is correct to say that there's a high probability that the true population value you are interested in falls in this interval. In general, our mean difference is the signal we're interested in. So which 95% confidence interval are we going to calculate? In this class, the 95% confidence interval of the mean difference. Now, here's an example of one. I have a 95% confidence interval of the mean difference that goes from -10 to +10. Now, right now, you're probably asking, what does this mean? What's present in this interval? First, the center of the interval provides your sampled mean difference. So in this case, it's 0. I am making these up, so the numbers work out very nicely for interpretation. Additionally, you have the spread of the interval, where you have your worst case scenario and your best case scenario for your hypothesis. When I calculate my mean differences, larger mean differences support my hypothesis. That's how I construct mine. So for me, my lower limit is my worst case scenario. So in this case, the worst case scenario is a 10-point mean difference that goes against my hypothesis. And my best case scenario is my upper limit, that positive 10. So best case scenario, there's a 10-point difference in favor of my hypothesis. So we know the center, we know the spread, and we also know p-value. If you don't understand p-value, ignore any time I mentioned p-value today. But if your 95% confidence interval of the mean difference includes 0, your p-value is greater than 0.05, and that means non-significant. If your interval of the mean difference, your 95% CI of the mean difference, does not include 0, that means your p-value is less than 0.05. It is significant. So I'm going to say this throughout the lecture. Small is significant. Not small is not significant. That's for p-values. Small is significant. Not small is not significant. What does that mean? Significant just means your two means are different. Not significant means your two means are not different. That is it. So small, significant. Not small, not significant. Where does the 95% confidence interval come from? It's actually pretty simple in concept. We apply a normal curve to our sampled mean difference, and we find the middle 95% of values in that normal curve, and we find the actual points that determine the lower limit and the upper limit. So in our case, you know, the normal curve is centered around 0, that lower limit is -10, and that upper limit is +10. So what matters so far? The true population value is likely in this interval. 95 out of 100 experiments, the 95% confidence interval will include the true population value. The mean difference equals the best estimate of the signal. This is the middle of the interval. We never really talk about that, but it contains your signal as well. It also gives you information about p-values. If the interval contains 0, your p is greater than 0.05. Not small is not significant. If the interval does not include 0, your p is less than 0.05. But for me, the most important aspect is that your spread provides the worst case and best case scenario of your true population value, and thus it shows you precision or control in your science. I think this is why a lot of scientists are unwilling to report their 95% confidence interval of the mean difference, or I should say were unlikely to support. I was reading Psych Science just the other day, and this process is more and more standard. But ultimately, the question you should ask when you're evaluating your science or the science of others is, am I making a good estimate of the population value, or am I making a bad estimate of the population value? And really, we're asking, do we have a precise or controlled estimate of the population value? All right, so how do you judge the precision of the spread? I said this was the most important thing. Well, what you should do, in my opinion, is convert your 95% confidence interval of the mean difference to a 95% confidence interval of the Cohen's d. Now, just a reminder, how do you do that? Essentially, you take your mean difference divided by the average of the two standard deviations. When we do the calculations, you'll see it's a little bit more complicated than that, but conceptually, we're right on. So don't forget your Cohen's d's interpretations. 0.2 is small, 0.5 is average or good in behavioral science, and 0.8 is considered large. So these interpretations will help you understand the precision of your effect size estimate. Now, ultimately, while in the previous lecture I did talk about how small effects can matter, when we are interpreting our own science or the science of others, we have to really recognize that a small effect is really saying that this intervention isn't great, that this vaccine doesn't work, that this really isn't the best use of our money or our time in terms of public policy or in terms of whatever. So from now on, when I talk about a small effect size, I want you to understand that I understand that small effect sizes can matter in very specific circumstances, like life or death matters. However, In terms of science and scientific interpretation, I need you to realize that a small effect size basically means your study didn't work and isn't showing much value in terms of whatever you're trying to show. So we have 0.2, 0.5, 0.8. 0.5 is the average effect in the behavioral sciences. 0.8 is a large effect. Just keep those in mind. So here we start. We have our 95% confidence interval of our mean difference. It goes from negative 0.5 to positive 0.5. I don't know what that means, right? Let's say it's a 5-point scale. That's like a half-point difference in either direction. Is that good? Is that bad? I honestly don't know, because it depends on your standard deviation, and I need to convert these mean differences to Cohen's d. And for today, I'm going to make the math super simple on myself and on you as well, because both of our standard deviations are 1. So now our 95% confidence interval of our Cohen's d is also negative 0.5 to positive 0.5. Do not be confused. The standard deviation is not always 1. I'm just making the math easy on us today, because we have to really think about, is this a precise interval? So what do we know right now? Well, it includes 0. So we know our p-value is greater than 0.05, not small, not significant. Okay, so our p-value is not small, it's not significant. We know our mean difference is 0 because I made up the data, and that's the center of the interval. Worst case scenario, we have a medium effect in the opposite direction than the direction I predicted. And my best case scenario, there's a medium effect in the direction I predicted. Do we have precision in our estimate? And remember, I always set up my confidence intervals so that positive numbers, I always set up my mean differences and thus my confidence intervals so that positive numbers indicate support for my original idea or hypothesis. Is this interval precise? Let's see. So ultimately, I've mapped out sort of Cohen's levels of small, medium, and large, and I've added a no effect in the middle to see, like, what do I know? What do I know about the size of this effect? Green means it's a possible interpretation of the true population value that I'm seeking, that true population mean difference. So green means it's possibly any one of these, and red means it cannot be this specific interpretation. So what I know about this mean difference is that it is not a large mean difference for or a large mean difference against my hypothesis. So what do I know? This possible population mean difference that I'm interested in, the one I'm trying to make inferences about, could be an average effect in favor of my hypothesis, a small effect in favor of my hypothesis, no effect at all, a small effect against my hypothesis, or a medium and average effect against my hypothesis. Average and medium are the same. I'm using them in there for you just to get used to it, okay? But ultimately, do I have precision here? What do you think? I mean, the effect could be anything from an average effect for to an average effect against or a medium effect against. No, we don't have precision. This could support or disprove my hypothesis. It could be nothing. I just know it's not large. That's not a precise understanding of the true population effect. What I do know is it's not large in either direction. That's not very useful. So I don't have very good precision. And right now I'm saying I have no idea what that true population effect that I'm trying to understand, trying to find, I have no idea what that is. My spread is too large for me to really say I have good science. What I do know is that I should redo the study and remove any source of error that I can find. New problem, I'm going to go through it a little bit quicker. Our 95% confidence interval of the mean difference is from negative 0.01 to positive 0.01. Just assume a Likert scale from me. My average standard deviation, which I'm making up, is 1 so that the math's easy. I'm just making the math easy for us. If it was 2, the next confidence interval would change. Our 95% confidence interval of the Cohen's d is from negative 0.01 to positive 0.01. And I know my p-value is greater than 0.05 because 0 is in the interval. And ultimately, I want to interpret that spread. Now, let's interpret that in terms of effect sizes. That is extremely small. I'm going to call it small for right now, but it's either the smallest effect ever in support of my hypothesis or the smallest effect ever against my hypothesis. So here we have the same thing. This effect cannot be a medium effect against, a medium effect for, an average effect against, an average effect for, cannot be a large effect for or against my hypothesis. So I know the effect is not medium and not large. Well, this is important because I'm just telling you that if we have a small effect, it's basically nothing. So what I do know is that this effect is basically nothing. It could be a small effect for, it could be no effect, could be a small effect against. In terms of precision, what do you think? Do you think this is precise? Do you think it's not precise? And in fact, I'm struggling between two interpretations, so I'm going to show you them both. My first interpretation is it's okay prediction. It's crossing three of the possible interpretations. It's small, no effect, or small. And that's not as precise as I would like. In general, for me, I would like precisions to sort of cross only 2 pi slices, but that's not a hard and fast rule. And so, it's an okay precision. You know what I know? I know this effect doesn't matter in my field. I know that much. I know it's much smaller than the average effect, and I know I shouldn't use this in public policy, and I just don't know if it helps or harms people or if it supports or goes against my specific hypothesis. The second way I'm thinking about this specific effect is that 0.01 is 20 times smaller than small, right? So in many ways, is it fair to call this effect a small effect when it's 20 times smaller than small? And I'm arguing no. It's just Cohen doesn't have an actual value that is no effect. I just put it in there because logically, no effect is between a small effect for and a small effect against. But essentially, if you're really asking me, when I look at this data and my Cohen's d is 0.01, that is 20 times smaller than small. That's essentially nothing. So I'm going to argue that I do have precision. I know there's no effect here. So yes, I do have precision. If you have taken stats before, you've probably heard that you are never allowed to say that two means are the same. Now, this is true only when you're using p-value, but when you use the 95% confidence intervals, you actually can start to make a logical argument that your two means are the same. And I'd say we have a strong case that we can make that these two means are the same, as I was talking about in the previous slide, right? Ultimately, you will hear, if you're just using p-values, only p-values, when your p is greater than 0.05, you know nothing. But we are expanding your repertoire of statistics beyond p-value. You can make the argument that these two means are the same. I'd say we should make the argument that these two specific means in this made-up example are the same, where our confidence interval was from negative 0.01 to positive 0.01. So how do we do that? And this is what I would write for publication if an author would give me a problem. Now, I was reading Psych Science, and I really don't think that every single reviewer would object to this automatically. It's becoming more and more acceptable to put your confidence intervals of your mean differences in your papers, and it's actually becoming expected. So here's what I would write if I got a reviewer that was challenging my claim that the two means were the same. I would say the 95% confidence interval of the Cohen's d suggests, at worst, the true population mean difference is an effect that does not support my hypothesis, but this effect is 20 times smaller than what is considered a small effect in our field. At best, the true population mean difference does support my hypothesis, but is also 20 times smaller than what is considered a small effect in our field. It is logical to interpret an effect that is 20 times smaller than small has no effect at all. Thus, this confidence interval suggests that the true mean difference is practically zero. Notice the word practically there. That's what's going to save you. And while it is generally frowned upon to suggest a non-significant p-value is support for equal means or has support for equal means, the 95% confidence interval of Cohen's d clearly suggests that this is the best interpretation of this data. Period. Mic drop. Leave the room. Okay, a new example, the mean difference confidence interval from 1 to 2. We know the mean difference is 1.5. Assume a Likert scale, it's a pretty big difference. P-value is less than 0.05. 0 is not in the interval, right? Small is significant. So this would be a significant difference. The two means are different. The control and the experimental mean are different. Average standard deviation equals 1. Remember, I'm making these up. The standard deviation is not always 1. This is a day where we're thinking about interpretation. Our 95% confidence interval of the Cohen's d ranges from 1 to 2. Okay, let's see what this looks like on my pie chart. Well, we only have one interpretation. This is a large effect in support of my hypothesis. Do we have precision? Oh, definitely. This is the best case scenario. So we have precision. Next example, 95% confidence interval, the mean difference ranges from 0.01 to 2. Your mean difference is 1.005. Your p-value is less than 0.05 because 0 is not in this interval. 0 is outside the interval. We know our average standard deviation equals 1 because I'm going easy on us today. So our 95% confidence interval of the mean difference ranges from 0.01 to 2. Do we have a precise estimate? So we can see that we really wish we had a more precise estimate. What's important here is that I do know something. I do know that the evidence generally supports my hypothesis. There is a difference. But what I don't know is that difference small? Is it medium? Is it large? I don't know. And in fact, if you look at my limit, which was 0.01, that's another effect that's 20 times smaller than small. So in essence, all I know is that the evidence does not disprove my hypothesis. That's all I know. It does support my hypothesis that there's a difference, but I don't know if this difference is important. I don't know if it's the largest difference in the world or a meaningless tiny difference that's 20 times smaller than what we consider small. So how am I going to judge this in terms of precision? I'm going to say it's it's okay. It's okay because I know something. I know there's a mean difference in the direction I predicted, right? I do know that. And that's okay. And in fact, this is all a p-value tells you. When you are calculating p-value, all you know is there's a difference, but you don't know how small that difference is. And this is why I find p-value a little frustrating. A lot of people think, oh, my p-value, it tells me that there's an actual difference, that it matters. No, actually it just tells you that there's a difference. The difference may be as tiny as a 0.01 difference or as massive as I think it was, a 2 standard deviation difference, which is massive. Largest difference that I would have ever observed in my research. So it's okay. That's part of my thinking. It's okay because part of me has been brainwashed by the p-value logic thinking. I know that there's a mathematical difference between these two means. It just may be the tiniest difference in the world or the most massive difference in the world. Really, I should rerun this experiment, tighten up my measures, my methods, so that I can get a more precise estimate of the true population value. When it comes to the question, do I have a good handle on the true population mean difference or Cohen's d? No, I don't really. I know it's not 0. I know it's greater than 0, but that's all I know. Now, going back to my argument, 0.01 is 20 times smaller than what we consider small. Isn't that, for practical purposes, a no effect as well? So that's the way I like to think about it. That makes me say, ooh, yeah, my p-value is less than 0.05, but I don't know if this effect is, for practical purposes, a 0 effect, a non-existent, non-important, non-relevant effect. So, or it could be anything to the most large effect I've ever observed. So my second thought on it is like, ooh, and you can see I changed that little green arrow down there because it's basically a no effect. It's not technically a no effect, but it's right up to the edge. So is it precise? Barely okay by my standards of logic. Now, if you're following along and you agree with me, what you're agreeing with is that the logic of the p-value isn't that great. You don't want to make your inferences like that. Like, oh, I'm happy when I find that the true population mean could be practically nothing to practically massive. Like, if you're agreeing with me right now, you're actually agreeing that p-value is kind of a poor way to think about your data. P-value alone is a poor way to think about your data. In fact, if I ran the world, I wouldn't even have us focus on p-value. I'd have us focus on precision, right? Like using my little pie chart, I would say, okay, if your experiment isn't only 2 pies precise, then you don't really know anything. And I would also define a small effect and a practically non-existent effect. I would say 0.1 is practically non-existent. Two means differ by 0.1 standard deviations. That's the Cohen's d of 0.1. That's practically nothing. That doesn't even matter to me. And I mentioned this, I didn't realize I would bring out all the logic, but I'll just restate it. This is essentially all a p-value tells us, is that practically our effect may be meaningless and worthless, or it could be massively large. It could be anything from practically meaningless, worthless to massive and large. That's all we know with a p-value. And you should start to distance yourself from p-values, especially those of you that want to become real scientists in the world, because at least in psychology and in many parts of cognitive neuroscience, people are moving away from this antiquated understanding, this black and white understanding of our experiments that we get from p-value. But this picture right here represents what we know with a p-value. While technically it's not an effect, it's approaching it. While technically it's not no effect, the effect is so close to no effect that it basically practically might as well be no effect, or it could be a large effect. When you see that your p-value is less than 0.05, that's all you really know. And larger p-values don't save you. I will prove that in a slide or two. So what's my goal with this class, or even with today's lecture? I want you to think about your data. I get so many students that get frustrated with me sometimes because I'm not okay with them just finding a mathematical answer, right? And not thinking about it. Oh, I have a p-value, so it's significant. And everybody else around me is saying that means I found something important. It's not everyone else. There's a small group of us that are we're less on the fringes now, but are like, ooh, that's your only method of interpretation? How big is your effect? What is the range of the effects that you think exist in the population? How good was your science? These questions are questions that frustrate some of my students because they're like, this is a math class, it should have an answer. But no, this isn't just a math class. We don't do research just to publish and to put something on a resume. We do research to make the world better, to make medicine better, to make social policy better. Right? We need to actually think about our data. We are talking about the difference between life and death. Also, if I get you on your poster session, I want you to be able to talk to me about precision of your effect size estimate. Now that effect size could be your mean difference. I'll be okay with that. I'll help you work through those discussions. Or it could be your Cohen's d, which I think is the better method. I just checked out the most recent issue of Psych Science yesterday. I was reading, I try to read an article a day. I'm failing, but every 4 days I read an article, I guess. And I've started to see, I'm so grateful, I've started to finally see folks are reporting 95% confidence intervals, not just of the mean difference, but also of the Cohen's d. So really, other people are agreeing with me. I myself am trying to pick up my research productivity. And so I'm relearning what's what's kind of standard in the field, and a lot of things have changed in just 15 years. Not 15 years, excuse me, 10 years. A lot have changed since I received my PhD in 2011, which is 9 years, I guess. But I want you to know that I'm trying to get you to think about your data because it matters, right? Like if you're doing research, I want it to matter. I want it to change lives. I don't want it to be about your resume. I don't want it to be about your project, right? Just getting a grade. I want it to be because it matters. And if you really can't, I probably have the moral high ground right now if you want to argue with me, but I don't think you're going to want to argue with me. I think you're going to want to join my team and go ahead, read Psychological Science, the best journal for very innovative psychological research, and see what we're doing. Also, that's where a lot of neuroimaging stuff is published as well. So cool. Why do you need to think about your data? Our fields, neuroscience and psychology, have problems. I'm going to start with neuroscience because I think it's kind of easier to pick apart psychological science in some ways. A lot of neuroscience studies, particularly with animals, have ends that are just disturbingly small. And people would often reason, oh, well, with small sample sizes, you're less likely to find a significant difference. Except this is not what people are finding. People are finding that these small sample sizes are making it very much more likely to find a significant difference. If you want to read the math on this, if you're a bit of a math nerd, read this paper, Power Failure While Small Sample Sizes Undermines the Reliability of Neuroscience. Read that paper. Psychologists, you probably want to read this paper over here, Estimating the Reproducibility of Psychological Science. So if you read that abstract, 36% of replications had statistically significant results. 47% of the original effect sizes were in the 95% confidence interval of the replication effect sizes. Essentially what we're finding there is, let's just take the p-value stuff. 36% of replications had statistically significant results, meaning 2/3 did not. These problems exist not just in psychology. They exist in biomedical medicine. You can find reports on that as well. There's a paper that suggests that most medical research is false by John Ioannidis. But what I need you to realize, and what I need you to realize as people, as majors, is that even the stuff you're reading in our class could very well likely be wrong. Okay? Very well likely be wrong. And so it's hard for me as a scientist to trust the papers of others. I'm reading what's going on. I'm reading what's out there, but I'm also like, I know what's going on in the field to a very personal standard because I joined this field. I had to publish stuff to get a job. You know, to get a job, you have to get published to get promoted. That's a lot of pressure. People have done worse with less pressure. And I think as a field, a lot of my colleagues that work at the bigger schools, they need to get millions of dollars in grants to keep their jobs. And that pressure doesn't really go away. And even while they may be able to keep their teaching job, that's not what they want to do. So they very much can lose the very thing. Like if you had 2 jobs at your one job and you loved one, they love research, and you hated another. A lot of hate is probably a strong word, but very much dislike research or dislike teaching compared to their research. Isn't that a lot of pressure on them to keep doing the thing they love so that they don't have to do the thing that I love, right? Like I think that's really interesting to understand these pressures that exist in our fields. And what I need you to do is not be subservient to every PI, to every person, to every article you read that's published. A published article based on these accounts is very likely to be false. So one of the things I want to show you is a recent demonstration that shows how bad p-values actually can be. So in this demonstration, what Jeff Cummings used, Dr. Jeff Cummings, in a paper called The New Statistics, published in Psychological Science, the best journal in the field of psychology, showed that, p-values don't really lead to truth as often as we would think. Now, in order to understand this graph, you have to understand something called a Monte Carlo study. Now, with a Monte Carlo study, instead of actual population, you create a population in a database. So it's just numbers with values. So in this case, actually create two populations. One's a population in the control group, and one's a population for the experimental group. And what we know is that those two populations have a population mean difference of 10. So you could imagine that this will have 4 billion numbers to represent 4 billion humans. It was probably more like 100,000 numbers in each population. That's actually pretty easy to do and well within the range of computing power today. But you start with a known population of data. Now, this population has a population mean difference of 10, and both of those smaller populations, the control group population and the experimental group population, have a standard deviation of 20. So when you take 10 divided by 20, the effect size is 0.5, and that's why Jeff Cummings chose it. So this is an effect that's the average effect size in our field, or what we consider a medium effect in our field. It's basically saying this meets a standard in our field that we all use. So you have this population, these known factors. And what you need to realize is that there's a real population difference, so your p-value should be significant, and your confidence interval should include that real value of 10. The other thing you need to understand is that how do you use that population, that Monte Carlo created population? Well, what you do is you just randomly sample a set number of observations from each of those populations, and then you run traditional statistics. You calculate your p-value, you calculate your 95% confidence interval of the mean difference. Okay, so what sample size did they use? They chose 32 because 32 is the average sample size in our field when Jeff ran this study. I would say it's drastically gone up. I just read a paper with about 2,000 participants just the other day. So you sample one 64 participant or observation experiment, and you run your p-value, you run your confidence interval. When they did that the first time, they found a p-value less than 0.05, and the confidence interval included 10. Both of those led to truth. And then what Jeff Cummings did was repeated it just 25 times for this demonstration, but you could repeat it 1,000 times. You could easily repeat it 1,000,000 times if you gave your computer probably a couple hours. So let's just focus on p-value right now. Truth for this specific problem, there is a known population mean difference of 10, effect size of 0.5. Truth is p-value less than 0.05. How many of these p-values led to truth? The first one did, the second one didn't, the third one did, the fourth one didn't, the fifth one didn't, the sixth one did, the seventh one didn't, the eighth one did, the ninth one, that one's a toss-up. People disagree on this, so I'm going to throw out the ninth one because 0.05 for me, less than 0.05 is significant. For other people, equals to 0.05 is significant. But that's a rare case, but I'm going to throw that one out when we're estimating this p-value stuff. The 10th one didn't, the 11th one did, the 12th one did, the 13th one was close but didn't, 14th did not, drastically did not, or did and did not. So of these 25 Monte Carlo simulated studies where the population mean difference was 10 and the standard deviation was 20 and the effect size was 0.5, what percentage of them led to truth? Based on my counting, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. 11 out of 24, excuse me, because I left out the 0.05. Found truth. That's less than 50%. We're better off flipping a coin than using p-value to test our hypotheses in psychology. This should really question everything you've learned, especially when you look at your textbooks, and most of their conclusions are based on p-value. So I think it's really important to start being a skeptical scientist. Don't just trust stuff because it's published. Don't just trust something because it's a p-value. And also, what this shows is that you're going to have to develop an understanding of the machine that drives science if you want to be a scientist. That means statistics. That means methods. That means anything in those realms. Let's look at the 95% confidence interval of the mean difference. How many of them found truth? And that means included the mean difference of 10. The first one, the second one, the third one, the fourth one, the fifth one, the sixth one, the seventh one, the eighth one, the ninth one, the 10th one, the 11th one, the 12th one, the 13th, 14th, 15th, 16th, all included truth. The 17th did not. 18th, 19th, 20, 21, 22, 23, 24th did not. 25th did. 23 out of 25 of your 95% confidence intervals of the mean difference included truth. And if you look at these intervals, though, what do you notice? You notice that the precision is poor. The precision is poor in, I really wish we could, well, okay, so the mean difference, we convert these, we can easily actually convert these to Cohen's d. So that 0 is a Cohen's d of 0, so that's great. That 10, that's your Cohen's d of 0.5. That 20 is a Cohen's d of 1. And so really you're going to have to that point next to 1, right? So that's going to be small, medium, and large. 0 is non-existent, small. Okay, a few points out, like 1, 2 ticks. You're not going to see it, but you have to count the 2 ticks. That's going to be the small line, right? So let's look at this at the precision of the first estimate. Medium to massive. Second estimate, small to massive. Third estimate was actually really good, right? Well, not really good. It was okay. It's basically medium-ish. Yeah, medium to large. And that's about the best you're going to get. Here, you got medium and opposite to medium in favor, essentially. Maybe a little bit more precise than that. But when you're looking at the precision of these intervals, what you're noticing is we don't have great precision. If we had great precision, we're going to be more likely to find truth. None of these intervals are really precise. The most precise is that third one. And in that one, you have both a significant p-value and a confidence interval that includes truth. How do we increase precision? We increase sample size. We increase the quality of our methods. We increase the standardization of our methods. It's that aspect that I want you to focus on. Yes, the 95% confidence interval gives you truth, but also it tells us how bad our estimates of truth and precision are, of the mean difference of the population are in the field of psychology with our standard practices. This is unacceptable. This is the thing that we need to improve. You can see it right here, but nobody really talks about it. Increase the precision of your study. Increase the precision of your measurement. Increase the effectiveness of your methods. Increase the number of participants. Because while clearly 95% confidence intervals of the mean difference provide truth, way better than p-values do. What you can see in an estimate of how we do our science is that our intervals are very, very imprecise. And I would say show a lack of control. And that is not what good science is known for. This is a message to you as the future of the field or whatever field you go into. I think these lessons can apply broader than just p-value, but do better than those that came before you. We were trying. I'm not going to lie. I know I'm not mad at my advisors. They didn't know any better, right? We're just trying to do things to make the world better, and we're using stats because that's what we have to do. That's what we were taught to believe to find truth. But now that we know better, we can do better. So for each of these 95% confidence intervals of the mean difference, tell me what you know. Sampled mean difference, the p-value, and the precision. Assume standard deviations of 1 for all conditions. And here are your 4 confidence intervals. Tell me what you know.